Hi, and welcome to our first lecture on Mendelian heredity. Let's get started. So first, let's define heredity. Heredity is the passing of traits from offspring to parents, and it's done through sexual reproduction. And we learned last chapter that sexual reproduction is a great way at combining and rearranging genetic or promoting genetic variation. This is in addition to the process of meiosis, which recombines genes through the process of crossing over and independent assortment. So you get a lot of genetic variation here through these three processes. And that's really important to how heredity is going to pass down traits from one generation to the next. So there's four key topics and concepts in this chapter. Today we're going to look at genotypes and phenotypes, and we might introduce Punnett squares. There's the three laws of inheritance as established by Gregor Mendel, hence Mendelian Genetics. You kind of are familiar with two of them already, so that's pretty nice. And then lastly, we'll do pedigree charts, but that's not today. All right, let's get started. Before we kind of begin our discussion on Mendelian heredity, I wanted to remind you guys of the Bohr model, which is something you learned in 7th grade. So the Bohr model is a very simplistic representation of an atom that tells you some pretty easy information to get it. So, for example, you have protons in the nucleus, neutrons in the nucleus. There's 6 of each. And then if you count up these little electrons on the outside, you'll see that there's 6 there, and there's in 2 energy shells. So it's kind of a simplistic model, but it does some things bad, like it gives the impression that electrons orbit the nucleus like planets orbit the sun, but that's not really how it works. A more sophisticated model is something called the Vesper model, where you can actually see the electron cloud formations, what those look like, which are probabilities of where these electrons will be. But this doesn't tell you the kind of information that you might get from the Bohr model. So there's pros and cons to both. The idea being that the Bohr model is fairly simplistic, but it gives us some really good information for foundational understanding of how atoms work. So the same thing is going to be true for Mendelian genetics. It's a simple model that gets at the basics, but it's not going to answer every question about heredity. But we'll get there. Okay, so before we begin, let's look at some familiar vocabulary. An allele, this is one of many alternative forms of a gene. Remember, a gene is like a category, like hair color or eye color, and then the allele is actually blue eyes, brown eyes, red hair, brown hair, that kind of thing. A gene is going to be your segments of DNA that carry hereditary instructions. Traits will be a distinguishing quality or characteristic that can be passed from one generation to another. And then lastly, your DNA sequences have matching genes, but may differ in some or all of the alleles, because you're getting two alleles for every gene. One came from mom and one came from dad. All right, so our definition of phenotype and genotype. The phenotype is the outward appearance, the expression of a trait. So once your DNA has gone through transcription and translation and it creates a protein, how is that protein going to be translated, or how's that protein going to appear to other people? How is that going to appear to yourself? What are you going to look like? What are you going to act like? How are you going to feel? What will you sound like? What will you smell like? Some of these things are things that you can physically observe, but phenotypes aren't just limited to physical descriptions. It can also be things like how well your cells metabolize lipids, or how well your stomach cells produce hydrochloric acid. So there's phenotypes is basically just any description of what your traits are doing. The phenotype is a result of the genotype, and the genotype is the combination of alleles a person has for a particular gene. For example, if the allele, for example, well, we'll get to the examples in a second. One quick note about genotypes is that if the allele is dominant, we use a capital letter, and if it's recessive, we use a lowercase letter, although it is the same letter. All right. Tricks to remembering the differences. Genotype starts with G, and so does a genetic code. Phenotype starts with PH, and so does physical description. So, for example, big G, little g, that would be a genotype. Big H, that's a genotype. Green eyes is a physical description. Curly hair is a physical description. Those are phenotypes. How many alleles do you have for every gene? You have 2. One came from mom and one came from dad. We represent alleles with upper and lowercase letters, but remember, it's just that this letter is representing a whole sequence of an allele, of a chromosome. So it's really just shorthand for maybe 1,000 or 10,000 or 100,000 nucleotide bases in a sequence. So when we say that this chromosome part, let's say that this is an allele for curly, wavy hair, This little h really represents a tremendous amount of DNA sequence. And that sequence is there to produce a protein which may or may not result in the trait that you're describing. And if the protein is just slightly different due to a change in the sequence of nucleotides, it can result in a slightly different trait. And that's how we have that kind of genetic variation between members of the same species. Okay, how many possible two-letter combinations can you make using the following alleles for hair texture? We have a big H and a little h. And so basically there's only 3 combinations. You can do big H, so this would mean that each of your parents passed down to you a dominant H. It could be that your mom gave you a big H and your dad gave you a little h, or it could be that you both got little h's from your parents. So your mom and your dad passed down 2 recessive alleles. Now you might be wondering, there's also little h, big H, but that's still the same combination as big H, little h. It's a different different permutation, but it's the same combination, so we're not going to worry about that one. All right. Okay, 3 possible genotypes. Those 3 versions, big H, little h, and big H, little h, they have names. When you have 2 dominant alleles, it's called homozygous dominant. Homo meaning the same, and zygos meaning of the zygote. So this is a zygote that has the same dominant alleles. 2 little h's mean you have a homozygous recessive, which is a zygote that gets the same recessive alleles from their parents. And then heterozygous is different. Hetero means different, and zygos is a zygote, so 2 different alleles. One important thing to note is that whenever you have 1 big H, that will result in the dominant phenotype. In order to have the recessive phenotype, you need 2 little h's. And that kind of gets at the heart of what a dominant and recessive allele is. Every organism has traits, and they're each represented by that 2-letter code, the genotype. Some alleles are quote-unquote stronger, and we call those dominant, while others are weaker. So the definition of a dominant trait is a trait that requires only one of the alleles to be inherited, and that will cause the dominant phenotype. So for example, a regular thumb will result as long as you have just one allele that codes for a regular thumb. But if you want a hitchhiker's thumb, you need to inherit both alleles that code for hitchhiker's thumb. So this makes this recessive, and just in having one of these traits is, or one of these alleles will allow you to be have a dominant trait. And conversely, a recessive trait is one that requires both of the same allele to be inherited. So here's your hitchhiker's thumb again. In order to have a hitchhiker's thumb, you need both alleles to code for the hitchhiker's thumb. Here's some other examples there. One way to think of this as an analogy is if you think of a piece of glass that has 2 sides, an interior and an exterior side, you only need to paint one side in order for it to be colored, to be a piece of painted glass. You can paint both, but one side will do. In order for the glass to be transparent, both sides must be clear. So that's your recessive trait, where both sides have to be clear. This is sometimes it's thought of as a loss of functionality. And what I mean by that is that a recessive trait can be a result of a broken protein, so to speak. So when you inherit one broken protein, then it doesn't do anything, and the other working protein, the other working allele that codes for a protein, that's going to take over, and that will be dominant. But, well, for example, so if you have MC1R, and its job, which is a protein, and its job is to get rid of red pigment, when it isn't working, you get a buildup of red pigment in your hair follicles. But if you have one working protein, no big deal. It will still clean up all the red pigment. If you inherit 2 broken proteins, then you'll get red hair because it's not going to pick up any red pigmentation. So that's kind of how it works. It gets more complicated than that, but we'll leave that for another time. So how would you describe these chromosomes below? Your mom's chromosome, they're homologous. One came from her mom and one came from her dad. And we'll say that we're going to focus on this striped gene right here. So one has horizontal stripes and one has vertical stripes. And what that means is one of them is going to be dominant and the other is recessive. For your father, he has 2 of the same alleles. So he's going to have 2 dominant alleles. And we'll say B is, I don't know, we'll figure out, B maybe is brown hair. So when your mother produces gametes through the process of meiosis, 50% of her gametes will have this big B, and the other 50% will have a recessive little b. For your father, all of the gametes that he produces will have big B because that's all that he can give to those sperm that he creates. So let's see what happens when you combine those possible gamete alleles. So again, your mom, in one of her eggs, she'll have a big B, in the other one, she'll have a little b. And then for your dad, his only possible combinations are big B's, a dominant brown hair allele. So in this is called a Punnett Square, and its job is to represent the possible allele combinations in the children created when gametes combine. And if you're only looking at one trait, like hair color, it's easy to follow. So for example, in one possible gamete combination, you could have a big B that came from mom and a big B that came from dad, right? This big B here and this big B here. In this other possible combination, you would have the big B from dad and the little b from mom. So you have a heterozygous genotype. Here, again, same combination as the one above it, the big B here and the other big B here. And then right here, it's very similar. You have the little b and you have the big B from dad. So there's 4 possible combinations, 2 of which are the same in this scenario, and these 2 are the same as well. If you'll notice, they all have a big B. So they're all children in this scenario are going to have brown hair. But let's switch it up. Let's say your dad didn't have big B, big B. Let's say that he had a big B and a little b. In this case, you'd have a homozygous dominant child. You could have a heterozygous dominant child. And again, there's another possibility for a heterozygous dominant child with a big B and a little b. But if you look at here, here's a combination where you have 2 little b's. So from 2 brown-haired parents, because mom and dad both have brown hair, they could both pass down this little b, and you would have blonde hair. So that's a kind of an interesting scenario where the child can look different from their parents. When summarizing the results of a Punnett Square, you look at something called the genotypic and phenotypic ratio. In the last case, we have 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive, and that's summarized right here. 1 big B to 2 big B little b, 2 big B little b to 1 little b little b. The phenotypic ratio is the ratio of the possible phenotypes. And because 3 of these will code for brown hair and 1 will code for blonde hair, your ratio is 3 to 1. Okay, so that's where I'm going to stop it today. We'll pick up with more Punnett Squares later on. If you have any questions, please let me know.